Hi, everyone, and welcome to the second to last week of cell and molecular biology. This time we're talking about the extracellular matrix and cell junctions. So this material covers the first half of chapter 20 in your textbook, so go ahead and glance through, read, take notes on page 683 to 702 if you haven't done that yet. And then next week we'll cover the second half of the last chapter in your book. So we're almost done with the semester. So a couple of announcements before I get into the main material, but then we'll talk about different types of extracellular matrix, the molecules involved in these different types, and then the functions and types of the junctions between cells. So first, a couple of announcements. So a lot of you have done the lab this week, so thank you guys for being so proactive. It's a nice walkthrough about some procedures. I think it also helps inform our global awareness activity on the STAP incident. So go ahead and do that lab. Please give me some questions, things you're interested in learning about. I am using that to guide my lecture for next week. So once you've done that lab, go ahead and start on your STAP assignment. There is a presentation in Moodle that covers the original paper that sparked that incident. And so that's also a nice example for your literature presentation slides if you have any questions about how those should look. So speaking of your presentation drafts of what you're going to say, and your slides are due either to me or in Moodle Sunday, April 19th, so this Sunday. The sooner you get those in, the sooner I can look at them and give you some feedback. I know the end of the semester can be pretty busy with projects and things, so let me know if you have any questions about that. The final version will be then next week, Sunday, April 26th. So this fulfills the speaking extensive blueprint requirement for this class, and then the global awareness credit will be fulfilled by this STAP reflection paper. So all of those readings are in Moodle. A little overview of the original paper is also available. So let me know if you have any questions about that, but you should have everything you need to do that assignment. Okay, so those are kind of the big summary projects for this class to make sure we're meeting all of our requirements. Otherwise, we have the quiz after this lecture, one more lecture next week, and then that's all the new instruction I have. So there will be some kind of final. It's not going to be a sit-down exam type, but I'll have more details about that next week. I can tell you that it will be due the Wednesday during what would have been finals week. So Wednesday, that last Wednesday in April, the 29th, I believe. Okay, again, at any point, let me know if you have any questions. I'm happy to chat about any and all of these assignments. I think you guys are doing really well, so I'm really proud of you guys keeping up on all of this stuff. So with that, we're going to talk about extracellular matrix, so the material between cells. It can either be really rigid and inflexible, like with these bone extracellular matrix. It can be a little bit more flexible, but still provide a lot of structure, or it can be a little less rigid, like in our connective tissue. So lots of different flavors of extracellular matrix, lots of really important functions in our bodies and in other organisms. So it's not just the different molecules that make it up and their properties, but there's also different ratios of cells compared to extracellular matrix. So in our bone tissue, for example, the ratio of cells, the number of cells is really low compared to how much matrix there is. Also things like cartilage, the number of cells is somewhat lower compared to how much material there is, whereas in connective tissue, there are tons and tons and tons of cells, so a higher ratio of cells to extracellular matrix. Okay, so we're going to talk about these different types of matrix. So they're all made of a few different classes of molecules. So some give them a lot more structure. Things like collagen, this is one of the most abundant proteins in the human body. These help give cells tensile strength, so to resist stretching. There's also elastins, which help give the cell a little bit more flexibility, so kind of opposing forces at work with these different structural proteins. There's also protein, carbohydrate, or polysaccharide complexes. So proteoglycans play a really important role in these extracellular matrices. And we'll also talk about different glycoproteins that help stick things together. So either stick cells to the matrix or stick cells to each other. And so here on our diagram on the right, we can see an example of some epithelial tissue, some blood vessels. So all of these different cells are connected via the extracellular matrix. So really important for how our skin is put together and important for integrating lots of different tissue types. All right, so we've gone from inside the cell, individual pieces, now to thinking about how different things are being put together. So we've kind of gone to the other end of the spectrum in this class. Okay, so the first protein we're going to talk about is collagen. Again, one of the most abundant proteins in the human body. It has a really unique structure. So when it's synthesized, it's the structural protein, so it's more linear. It's this long polypeptide chain. But in order to function properly, it gets combined into this triple helix, triple-stranded collagen molecule. So 3 of these need to fit together very precisely. They'll wrap around. And then once these triple-stranded molecules are formed, they can combine and aggregate into a larger fibril. So there's mechanisms that keep this from happening inside your cells, because you don't want this structure to be on the inside. You want it to be surrounding your cells. So if you want details on that, you can check out the book. But for our purposes, we're just looking at the structure. So if we cut one of these fibrils in half, we can see all those individual molecules, those 3-stranded molecules. These fibrils can then combine together to form larger fibers. So each one of these little black circles is the collagen fibril. Each one of these fibrils contains many of these 3-stranded collagen molecules. So their structure is extremely important to function properly in our tissues. All right. So if we take another look at how these collagens are composed, there's lots of different classes of these molecules, but the main formula for the amino acid sequence is that there's a glycine amino acid followed by 2 different amino acids, so proline, and in this case, hydroxyproline. So HYP is a modified amino acid, so this is modified in the ER, and for extra details, you can check out your books, but for our purposes, just knowing that glycine, another amino acid, and then a different amino acid. So it has this repeating structure. So you can see glycine, proline, serine, glycine, proline, arginine, glycine, proline, hydroproline. This glycine amino acid, different amino acid formula is very, very important for the ultimate structure of the protein. These are very small amino acids in terms of the size of the molecules, and this is really important for their interactions with this triple helix shape. So there's a question here on the bottom. Want you guys to think about just a little bit. So why are codon deletions in the genes more serious than in genes encoding other proteins? So if we had a gene coding for the collagen molecule, and one codon was just deleted, all right, so we took this proline out and sequenced it that way, or translated it that way. Why would that cause such a bigger issue compared to other proteins, say enzymes or other things? So So I encourage you guys to pause this video and think about it for a minute. And once you've done that, kind of come back and listen to the answer. So because these are structural proteins and they need to fit together so precisely, if we mess with this glycine, proline, or glycine, amino acid, different amino acid formula, we drastically change the structure of this amino acid, or the structure of this protein. So if it was a globular protein, as long as it didn't interfere with the active site, it might not cause such a big deal. But if you have one wrong amino acid in one place, you've messed with that entire formula. And so not only have you changed one part of this helix, but all three components, because they're the same and fitting together. You mess with the structure at the smallest unit, and because they aggregate, so I'll go back one slide. If we mess with this portion right here, it can't fit together as precisely, so the smaller structure is impacted. That then impacts every other structure and has a really important implication for these proteins as a whole. So with structural proteins and collagen specifically, mutations in these genes can lead to pretty dramatic disorders and diseases. Okay, so just thinking about amino acids and structure and why these things are important. Collagens, though there are many, and they're very important, there's also another kind of structural protein. This one provides more elasticity to the cell. These are called elastins, and they can kind of go between different conformations. So they are cross-linked at their lysine residues. If a cell undergoes some kind of stretching force, these elastins can stretch out and maintain the cell's integrity. When the cell isn't under that stretching force, they can kind of dissociate and be ready for the next impact. So over time, some of these molecules are lost, but they overall give the cell the ability to withstand some stretching forces. So really important when we're putting cells under lots of different conditions. And then in contrast to collagen as well, these contain no modified amino acids, whereas collagen had that hydroxyproline. All right, so collagen and elastins, very important structural proteins for the extracellular matrix. How are these combined with cells? Well, we have these really nice proteins that bridge those extracellular fibers with the cells themselves. And so one class of these molecules are called fibronectins. These are really cool molecules that will integrate with proteins in the cell membrane called integrins. So seen here in this diagram, these really interestingly shaped proteins. Not only are they in the plasma membrane of a cell, but they also interact with the cytoskeleton. So in this case, we have some actin filaments that are attached to these transmembrane proteins via some adapters. So we're kind of hooking the cell itself onto these fibronectin molecules. They almost have that antibody-like Y shape to them. These integrin dimers can bind to these portions on the molecule right here called the cell attachment site. And then a little farther down the arm of these proteins is the collagen binding site. So almost like those little kind of grabby toys, or like some kind of pliers or other tool like that. They're able to bind collagen fibers and also bind the cytoskeleton of the cell. So these fibronectin molecules are really important in maintaining the cell's connection to the extracellular matrix. These proteins can be quite large, so you can see in this image right here a microscopic view of these fibronectin proteins. Okay, so fibronectins are really important. Another series of proteins and other molecules are these proteoglycans. So carbohydrate protein molecules. These are some made up of core proteins that are linked to glycosaminoglycans, abbreviated as GAGs. So these are a repeating series of disaccharides linked to some core proteins. So you can see here in purple these core proteins. These are those disaccharides, those GAGs that are attached. These are really important for cell matrices for a lot of different reasons. So one function is that they provide this hydrated space between cells. So especially where there's lots of movement happening, so in things like joints, it's really helpful to have this kind of fluid-like substance that can allow a lot of movement and things. So when we have these proteoglycans, it can retain a lot of water. It has this really viscous quality. They can make these huge structures that look like this and hold on to a lot of water. So these proteoglycans are really important in things like joints because they provide the hydrated space. We can also mix and match these different things so we can create lots of different types of proteoglycans. You can see some of the examples here and here. The other things they can do are binding secreted growth factors, so influencing the signaling that's going to cells. They can also impact cell migration in a couple different ways. So another really important class of molecules that are involved, that are part of the extracellular matrix between our tissues and in our tissues. Okay, so we talked about collagen and elastin, fibronectin, and proteoglycans. And now we're thinking about how cells are put together. So it's important for certain tissues to be held together very tightly, especially with our skin and different epithelial cells, cells that move, etc. So thinking about how different cells are held together or torn apart, they're mediated by many different types of protein-protein interactions. So this one's not in your book, but we'll chat about these different examples here. So for this first one, often involved in different immune responses, but specifically these NCAM proteins form these loops, and they're able to hold different kinds of cells together almost like a zipper. So these proteins go through the cell membrane. They associate with another cell that has the exact same protein sticking out of its cell, and they can zip together to connect. Cadherin molecules are another class of molecules that allow cells to recognize and bind to each other. We'll talk about those in a few minutes. These are two examples of things that are the same that are recognizing each other. There's also different kinds of interactions where selectin molecules can recognize some kind of glycoprotein on another cell. So we have some recognition events going on between different sets of molecules. And then here we have another kind of CAM, or circular loop-shaped protein. And these, instead of interacting with other CAMs, are going to interact with a class of proteins called integrins. We'll see examples of integrins in another slide or two. So these different classes of proteins all mediate cells interacting with each other. Sometimes it's to help stick together in a tissue. Sometimes it's selecting and recognizing different cell types to each other. And all of these work together to organize multicellular life. So the fact that we're able to make up tissues in different pieces is based on these protein interactions. Bundling of neurons to each other, these NCAM proteins are what allow different neurons to bundle together. And it also helps mediate cell movement. So not only things like leukocytes moving around, but also in development, as you have different pieces of the body kind of developing, different cells moving around, these different proteins play really important roles with that as well. Okay, so thinking about development, people who study development can look at these different classes of molecules. And when we look at cadherins, we know these are extremely essential for normal development because when researchers deplete the mRNA that codes for these proteins, so the cell's unable to make cadherin proteins, this is what a normal organized blastula would look like. Without these cadherin proteins that mediate cell interactions, there's just no organization. This is not going to develop into a healthy embryo. So extremely important for development of multicellular organisms. So I'll back up to that summary slide. So cadherins, again, were some of those similar molecule recognition systems. E-cadherin stands for epithelial cadherin, so the letter in front of it will kind of dictate what tissue type it's part of or its function. And we also have selectins and integrin proteins that we'll talk about here. So cadherin is important for development. Integrins and selectins also help mediate cell movement and cell recognition. So if we think about white blood cells moving around, very important for our immune response to inflammation and diseases. And so we want leukocytes to go where they're needed. And part of that movement is mediated by these different recognition proteins between cells. So this leukocyte's kind of floating around, almost like a tumbleweed, just kind of rolling along the endothelial cells. Once it comes to a point where it will need to go to a site of inflammation or an infection, it will start to recognize the proteins integrated in the cell membrane. So on the leukocyte itself, it has these selectin molecules, these selectin proteins that go through the cell membrane and interact with these glycoproteins over here. So the specific carbohydrate molecules will mediate that interaction. So these selectins recognize these glycoproteins in the endothelial cells. It will kind of stop itself from rolling farther. Once it gets close enough, it will recognize another class of transmembrane proteins, and those are those CAMs. And the CAMs, in this case, on the endothelial cells will bind integrin dimers on the leukocyte. And so these binding events will then mediate movement of that leukocyte to the site of inflammation. So selectins kind of recognize that initial point to stop, and then integrin recognition events, integrin binding with these ICAMs lead to that recognition event. Okay, so that was a summary of the different ways that cells can recognize each other and stick together. And the last topic we'll cover today are the junctions between cells. So all of this has been focused on animal cells. Plant cells do it a little bit differently since they have cell walls. So they have junctions as well, they're just a little bit different. But for the purposes of this week, we're focusing on animal cells. So the things that junctions places of interactions between cells, the different functions they have. It allows for communication, so things can travel back and forth. It allows separation between different cell layers. So endothelial cells are good examples of these different types of junctions, the different polarity of tissues and how junctions can mediate that. It also helps, importantly, exclude things that need to be excluded. So another aspect of having a cell membrane, you want to keep certain things out, you want to keep certain things in. And junctions between cells can help mediate that as well. And so the different types, there are some that help cells stick together. There are some called desmosomes and hemidesmosomes, and then there are gap junctions, so named because you can actually see the gaps between cells right here. So adhesive junctions, we have focal adhesions. Focal adhesions allow cells to bind to the extracellular matrix. Adherens junctions have certain proteins associated with them. Tight junctions are named tight because they're places where two cells are bound very tightly together, and nothing, not even liquid, is able to pass through. All right, we'll get to examples of these other types of junctions here in the next couple slides. So tight junctions, gap junctions, different places in between. Sorry, I should say gap junctions right here. Pardon me. Okay, so adherens junctions were one of those cell-cell connections that helps keep cells together. Again, found in a lot of epithelial cell layers in kind of belts around the cells. So I'll go to the previous slide. So these kind of blue tracks right here, these are good examples of adherens junctions. And so this kind of belt right here is connected directly to the cell right next to it. So these are really important for tissues that make shapes like sheets, like your skin layers, keep cells really tightly together. When you get blisters, that interrupts these different junctions. These are adherens junctions, and these specifically use those cadherin molecules, and they interact directly with the actin filaments, those microfilaments in the cytoskeleton in both cells. So adherens junctions make cells stick together. They bind the actin from different cells via cadherin molecules. Desmosomes